Good afternoon and welcome to the 2021 Charter Lecture. I'm Jack Hu, Senior Vice President for Academic Affairs and Provost at the University of Georgia. On behalf of the university, I would like to thank you for joining us for the Charter Lecture. The Charter Lecture series was established in 1988 to honor the high ideals expressed in the 1785 Charter that made UGA the birthplace of public higher education in America. Since 1988, we have brought leading voices from around the globe to our campus through this lecture series. Today, we hear from some of the UGA's most outstanding faculty members, the 2020 and 2021 Regents professors, Diane Marie Amann, Stephen R.H. Beach, Pejman Rohani, and Ronald Simmons. The Regents professorship is one of the university's highest recognitions, and these distinguished faculty members will be introduced fully to you in a moment. In delivering this year's Charter Lecture, our Regents professors underscore the UGA faculty's firm commitment to the high ideals of our Charter. I invite everyone watching this afternoon to direct questions to the panelists through the Q&A feature at any time in their talks. We will create the questions and post them to our speakers following remarks from all four of them during the Q&A. We're particularly pleased that President Moorhead is joining us this afternoon. President Moorhead is a 1980 graduate of the UGA School of Law and the first UGA alumnus to be named president in more than 45 years. He's also the Max Professor of Legal Studies in the Terry College of Business. Thank you for being with us, President Moorhead.
Thank you, Jack, and good afternoon, everyone. It's my honor to welcome you to today's virtual Charter Lecture, and I join you in recognizing UGA's 2020 and 2021 Regents professors. I would also like to thank the Provost's Office for sponsoring the Charter Lecture series. It's particularly fitting that we highlight these four individuals this spring, knowing that they have continued to advance the research mission of our university in the midst of what has been an extraordinarily challenging year. I believe our founders would be proud of the progress we have continued to make amid these difficult circumstances. As many of you know, Regents professorships are bestowed by the University System Board of Regents on truly distinguished faculty of the University of Georgia. Their scholarship or creative activity is recognized both nationally and internationally as innovative and pace-setting. The four individuals we have with us today are doing research that is among the best in their field, and we are very proud of their accomplishments. They set a bar for excellence that is helping UGA continue to rise among the best research universities in the nation. This afternoon, these professors will each present brief highlights of their research to us. Helping to introduce them today is Devin Gokhale, originally from Mumbai, India. Devin is a PhD student in ecology who plans to become a mathematical modeler and data scientist working in the field of public health. Thank you for helping us today, Devin. I'll turn things over to you.
Thank you, President Moorhead. It's my pleasure to introduce the 2020 and 2021 Regents professors. We will begin today with Dr. Ron Simmons. Dr. Simmons studies the processes by which social experiences become biologically embedded and influence mental, physical outcomes. His research suggests that social factors such as marital, work, financial, and social status variables impact biological aging and the development of chronic illnesses. more so than the effects of diet, exercise, body mass, and other known health risk factors. Next will be Dr. Stephen Beach, whose scholarship focuses on the way that community, family, and marital processes affect mental and physical health. His pioneering work and collaborations have shown that strengthening close relationships, including marriage and parenting relationships, can play a critical role in reducing depression and physical health problems. Third will be Dr. Rohani, who conducts research that introduces ecological perspectives to infectious disease biology. His work investigates the importance of spatial synchrony in the outbreak of measles and pertussis for predicting the extinction and eradication of these major childhood infectious diseases. We will conclude remarks with Dr. Diane Murray Amann. Her scholarship examines international criminal justice, human and child rights, constitutional law, and security governance. Her current research will produce the first ever book on the roles of women professionals at the 1945-46 war crimes trial before the International Military Tribunal at Nuremberg. Thank you all. I will turn the program over to Dr. Simmons now.
Hello, folks, and thanks first of all to the Provost for organizing this event. This is pretty exciting to be part of this. And thanks to all of you that are tuned in today. I am part of a group of sociologists and psychologists that are interested in the way that social circumstances, life events, social relationships impact people biologically. They get under people's skin and actually influence their health. I'm sure that sounds really abstract, and so what I hope to do today is make this more concrete by talking more specifically about a particular outcome, which is biological aging. So let me first of all share my screen. Okay, so explain differences in the speed or rate of biological aging, which may sound like a pretty dry topic, but when you get to be my age, you'll realize that that's really a very significant topic. All right, so let me note first of all what you already know, because it sets the stage for what we're going to talk about, which is that the human lifespan has been expanding tremendously, and that's pretty exciting. So in many countries, people live to be in their mid-80s on average, but it is the case that the health span just is not keeping pace. So people are living a long time, but those last few years are limping along oftentimes with several chronic illnesses that they're trying to manage. The National Institute of Aging in the last few years has decided that we need to switch strategies. If we're going to expand the health span, then what we're doing isn't working. And they suggest that instead of trying to cure each of the various chronic illnesses like coronary vascular disease, diabetes, and so on, we should be focusing upon the underlying roots of all of these diseases, and that is biological aging. Old age and the physical decline that it indicates increases vulnerability to all of the chronic illnesses that eventually kill us. And so if we could identify the factors, significant factors that influence the speed of biological aging, there would be the hope, the National Institute of Aging argues, that we can slow down aging and expand the health span. We may not live longer, but we won't have a several-year period of misery due to chronic illness. Okay, so in order to do that, we need a clear-cut measure of speed of aging. So let's talk a little bit about that quickly. So how do you measure pace or rate of aging? So the mechanisms that underlie biological aging are kind of known, but there's a lot to learn yet. But one thing is really clear, and that is that epigenetic changes are a fundamental component of the process. Epigenetic factors, you might wonder, what is that? Well, they switch genes on or off. They influence gene expression. They dictate whether a gene is upregulated or downregulated. So that means epigenetic processes are really, really important, because DNA just kind of lies there dormant and not doing anything. And if it's going to do anything biologically, it needs to be brought to life by epigenetic factors. One of the most pervasive and well-studied epigenetic mechanisms is methylation. When a methyl group attaches to a segment of DNA, it inhibits expression of that gene, especially if the methylation occurs in the promoter region of the gene. So I'm sorry, I'm get a little bit technical here for a little bit, but it'll be important and there'll be a payoff hopefully later. So methylation, when it appears in a segment of DNA, it tends to downregulate that gene. When it dissipates or disappears, then that gene tends to express itself. Now, importantly, since the 1960s, researchers have been aware of the strong association between age and DNA methylation in many sites around or throughout the human genome. So there are many places in the genome where the older you get, the more methylation tends to kind of to build up in those sites. And then you've got other sites where as age progresses, methylation declines. So you've got genes that are gradually turning on and turning off various parts of the body with age via this mechanism of methylation. So given this pattern, what people recognized a few years ago is that you could use the presence or absence of methylation tags at various sites on the human genome to make a prediction about how a person is aging. And this will be the last technical slide, I promise. So that's given rise then to epigenetic clocks. Epigenetic clocks are based on various sites throughout the genome where the level of methylation is strongly associated with age. A person's biological age then can be calculated by summing their methylation scores across the sites included in that index. Now, really this is pretty cool because it means that you can look at the methylation pattern displayed in a person's body, and by looking at which sites are methylated and which ones aren't, you can make a prediction about how old they are biologically. And then you can subtract their chronological age from that, and you've got a measure of the extent to which an individual is either accelerated or decelerated in terms of how fast they're aging. You've got a measure of how fast their biological clock is ticking, and that's pretty cool. I mean, it's there's nothing really quite like this. Importantly, these longitudinal, well, let me back up. If it was just cool, then that would be one thing. But it's more than just cool. It's really consequential because longitudinal studies of samples from countries throughout the world have demonstrated that there are that these indices, these methylomic clocks, or epigenetic clocks, are robust predictors of biomarkers of health, all the things that when you go in for your annual physical day assay, like blood lipids and blood sugar and kidney function and so on, it predicts all that stuff. Also, it predicts onset of chronic illness, it predicts your physical appearance, the extent to which you look old, your cognitive decline, and finally, time to death. I mean, so that's a pretty robust measure. Now, given that this is the case, and actually there are several of these, and the most recent ones, which we'll talk about in a little bit, are the ones that are really most powerful. But this raises questions regarding the causes of accelerated or decelerated epigenetic changes. So why do some people age faster than others? And you would undoubtedly say, if I asked you that, is genes, which you inherit from your parents. And you'd be partially correct in that about 40% of the variance between people is a function of the DNA that you inherit. But that means that well over half is due to some other things that we may well be able to control, and therefore could intervene in the speed at which someone is aging. Now, as social scientists, we're interested in the question, are social conditions and experiences, as well as lifestyle, a part of the story? And What we do is we draw blood from our study respondents, we calculate these biological clocks, and then we examine the extent to which socioeconomic, psychosocial, and lifestyle data collected over the life course of our subjects predicts their speed of biological aging. And so let me show you then, with the remainder of my time, what we found. First of all, our very first study just looked at the extent to which economic hardship would influence, might influence speed of aging. And you can see here, here's income, here is, these people are older than their chronological age, these people are younger. The numbers actually stand for years. And so if you're making approximately 10K a year, in our sample, you're about 2 years older biologically than you are within your chronological age. And if you're making 100K a year, you're about 3.5 to 4 years younger biologically than your chronological age. So having enough money to be comfortable is good for your biological aging. We then looked at neighborhood conditions, and if you ask the question, does a disadvantaged neighborhood impact your speed of aging? The answer is yes. It accelerates aging by about 9 months. By the way, in all this research that I'm going to be showing you today, we're controlling for things like diet, and exercise, and smoking, and so on. One of the most exciting clocks is the Grimm age clock. It's got 1,000 methylation sites. It is so good at predicting time to death that it's called Grimm age, named after the Grim Reaper. And you can see here the kinds of things that it predicts. And in our data, education, income, and marriage are significantly associated with decelerated Grimm age, whereas living in a disadvantaged neighborhood, discrimination, and financial hardship are associated with accelerated Grimm age. Differences in speed of aging appear to emerge early in life. We've known for a long while that childhood adversity predicts chronic illness in middle age. As a matter of fact, the relationship is so strong in the American Heart Association. If you look at their website, they describe childhood adversity as a risk factor for coronary heart disease. So our thought was, well, maybe this is because childhood adversity increases your speed of aging, and it does. So in their mid-50s, those who were had experienced significant amounts of child adversity were 4.6 years older than their chronological adult age. And using a different measure, POAM index, it indicated that they were aging at a pace of 1.15 years annually, or that would be about 430 days every 365 days. So speed of biological aging starts to get calibrated early in life and tends to be maintained over time. But here's the good news, is that when you look at people in middle age from about age 50 to 60, what we found is that during that decade, people are still showing a lot of change in their speed of aging, about a third increase by at least a year in their speed of aging, and about a third decrease. And so then you can ask the question, do changes in social and psychological factors impact these changes in speed of aging, for better or worse? And the answer is yes. And I won't go over all these findings. Let's just show you one. Like, if during middle age you fall in hard times and you have to move in with your adult kids, that's a very strong predictor of an increase in biological aging. That's very stressful. Lastly, we looked at incarceration, and we know that incarceration is extraordinarily stressful for people. They oftentimes come out with PTSD. They almost always have lasting poor health conditions, and they oftentimes suffer premature death. So our thought was, well, does incarceration itself tend to accelerate aging? The answer is yes. The effect is for men and for women. And it can be relatively short sentences of just a year or so. And the effect is really strong if you were the victim of assault while you were in prison, either at the hands of fellow prisoners or guards. I'll show you just one other thing. That is, the Grimm age scores, they correlate 0.32 between married or cohabitating romantic partners. So romantic partners tend to have similar rates of aging, which is really pretty amazing because there's no genetic effect operating here at all. So it appears to be the case that romantic partners share a life, and apparently this shared life somehow gives rise to similar rates of aging. This will be something we'll be investigating in the future. So, conclusion, takeaway message. The events and circumstances we experience over the life course influence, for better or worse, our speed of biological aging and, in turn, onset of chronic illness and the time till death. This line of research is in its infancy, but it would seem to have important policy and intervention implications, but this is not the time or place to sort those out. Instead, I will turn things over to my esteemed colleague, Steve Beach.
Thanks very much for that, Dr. Simmons. The kind of research that you presented showing the importance of social determinants of health provides the foundation for the work that I'll be presenting today. Building on that foundation, I'll be talking about ways to strengthen families using family-based interventions to reduce behavioral and physical health problems. Before I do that, I do just want to quickly say thank you to the outstanding research leadership team we have at UGA, to my students, to the external funders of the research that I'll be presenting, and the collaborators that I've had over the years at UGA. I've met just so many outstanding researchers here who've enriched my research and my life in lots of different ways. So I wanted to say thank you to them as well. Many of them I've met through the Institute for Behavioral Research. When I got to UGA, I was already very interested in marital relationships and strengthening marital relationships as a way of combating episodes of depression. I was working in a clinical context, so I was working with depressed individuals who had marital problems to show that we could substantially improve those marital relationships, and that improving their marital relationships had an effect on their depressive symptoms, a substantial effect. And I continued that line of research after I got to UGA, but my research also expanded in other directions, thanks to influences and opportunities that emerged at UGA. In particular, I became more prevention-oriented. I started wondering whether we could do more than simply react to problems that had already developed, and instead help strengthen families to head off those problems before they developed. And I also became more interested in health disparities, wondering about how we could help families living in more challenging circumstances who, for the most part, had not been involved previously in prevention efforts. So here's where we conduct our research at the Center for Family Research. And you can see we are heavily engaged in communities in rural areas of the state, with partnerships in those communities across the state. And that allows us to focus our prevention efforts on those who have been underrepresented in previous research on prevention. I also want to acknowledge that community-based family prevention research is inherently a team effort. It would not be possible without the support provided by CFR staff. And so here are some of the heroes of our research efforts at CFR, and I want to say thank you to all of them as well. The research program over the past 20 years at the Center for Family Research has resulted in a suite of 5 different interventions designed to promote strong families in developmentally appropriate ways. And you can see more about them at our website, which is at cfruga.edu. However, today, I'll just be talking about the Protecting Strong African American Families program and providing some recent results from that program. The program was developed by our program development team at CFR to meet needs of African American couples raising pre-adolescent and adolescent youth here in the rural South. It has 6 sessions. It's delivered in families' homes. It's expected to have direct beneficial effects in promoting positive family interactions and strengthening several facets of protective family processes. To generate the data that I'll be talking about, we recruited 360 families. We randomly assigned them to participate in PROSAAF or a control condition. And then we collected 4 waves of data over a 2-year time frame. The study was designed to test 4 different types of effects that are important in understanding the potential beneficial effects of family-focused intervention. First, of course, we were interested in seeing whether or not we could have direct beneficial effects on those facets of couple relationships that we were targeting. Second, we wanted to see whether we would find expected ripple effects, or what we call indirect effects, on parents in areas like health and mental health and sleep. And these are benefits that should follow from improvements in relationships and begin to address health disparities downstream. Third, we wanted to see if there were beneficial ripple effects for youth outcomes. And fourth, we wanted to see if PROSAAF could reduce the erosive impact of financial strain and other chronic stressors of the sort that Dr. Simmons was talking about, reducing their connections to problems for parents and youth. By reducing it, that's really what we mean by building family resilience. So we wanted to see if PROSAAF could be a program that would have an effect on enhanced family resilience. So the first question we wanted to address was whether PROSAAF could strengthen key facets of positive interaction in families, including satisfaction, communication, co-parenting, relationship confidence, spousal support, and protective parenting. And these are those 6 facets. As you can see here, and I'll walk you through this a little more slowly, but as you can see here, in all cases, there are significant effects of the program on these different facets. There's 2 sets of findings here that show you that the effects are parallel for husbands and wives. And what you can see is that in the PROSAAF condition, families are protected against the erosive effects over time. In the control condition, however, we see the expected erosive effects on protective family processes that emerge over time in these difficult settings. And you'll see that the patterns are very similar across all of the variables that we were looking at. So that set the stage for us to examine indirect effects and whether or not there would be ripple effects for mental and physical health outcomes and other beneficial changes for parents. So we found, as we already showed, we found the significant effect of PROSAAF on couple functioning. And then we went on to see these positive beneficial ripple effects on depressive symptoms, on sleep problems, on self-reported health. In these different domains, PROSAAF was having the expected beneficial ripple effects for the parents. What about kids? What about the youth? We also found significant beneficial ripple effects or indirect effects for youth outcomes, including enhancement of youth behavior and choice of peers, less early initiation of substance use, and improved positive self-control. So in each case, there was a beneficial effect that we were anticipating, which led us to then wonder whether or not we might be able to see those expected effects on resilience. Our first resilience question was whether or not we might see a reduction in the negative erosive effect of financial hardship on the couple relationship itself. And as you can see here, the impact of financial hardship, elevated financial hardship, was eliminated by participation in PROSAAF. So the PROSAAF program is shielding participants from what we would normally see, what we see in the control condition, where elevated financial hardship tends to reduce those protective processes over time. We also wanted to see if we would find a similar effect for protective parenting processes. And again, what we see is that PROSAAF eliminated the erosive effects of financial strain on protective parenting. And we didn't see the characteristic erosion over time from financial strain that we would see without PROSAAF. So given the success of PROSAAF in creating resilience to financial strain, we wondered whether PROSAAF might also buffer the impact of other stressors that put youth at risk, like neighborhood disadvantage or discrimination. So against neighborhood disadvantage, we saw that the answer was yes, and I'll walk you through this. Being in PROSAAF was associated with changes in parent communication and reduced arguing in front of kids, and that reduced the impact of neighborhood disadvantage on change in youth problem behaviors. So PROSAAF was buffering that effect on youth and increasing family resilience. We also found an effect of PROSAAF participation on buffering from racial discrimination, assignment to PROSAAF, increased protective parenting, as we talked about before, and that reduced the impact of racial discrimination experiences on youth depressive symptoms. So, to summarize, we found that PROSAAF had direct effects on several different facets of couple and family functioning that we believe should be important in protecting families. We found the expected ripple effects on a number of adult and youth outcomes. And we also found that PROSAAF was able to buffer effects, indicating increased family resilience. And all these results have been published over the past several years and have prompted us to want to follow up this sample to see what's happening subsequent to this intervention over a longer period of time. And so we are currently collecting more data on adults functioning, We're also looking at effects on biomarkers of health and effects on stress, effects of stress over time. So some of you are probably wondering whether or not we have cool data like Dr. Simmons. Now we do. And so to preempt the question that I'm sure at least some of you are wondering about, here's a preliminary analysis fresh from recently collected follow-up data from the PROSAAF sample, looking at indirect and resilience effects of PROSAAF on speed of biological aging, using the type of DNA methylation markers of aging described by Dr. Simmons earlier. And as you can see here, PROSAAF had the expected effect on relationship communication, and that resulted in an indirect effect on biological aging. That improved relationship functioning is also buffering the effect of financial stress on elevated biological aging. So it's improving these biomarkers of health as well. And that is a very quick walk through one line of ongoing research at the Center for Family Research. We have many more interesting results coming soon, including examining effects of family relationships on resilience to the stressors associated with the ongoing pandemic. So if you're interested, again, you can see us at our website. If you're interested in more methodological details or more background, the primary references for the material that I presented today are available on this slide. And so you can look this up later on. And with that, I'd like to turn control of the presentation over to Dr. Rohani, who will shift our focus to the ongoing pandemic.
Thank you very much. I'd like to start with a topic that is on many of our minds, which is infectious diseases. And this slide is a summary of infectious diseases, new ones, which we call emerging infectious diseases, and ones that we're familiar with, but nevertheless are increasing, escaping our control, are increasing in incidence and increasing in geographic spread, that we call re-emerging infectious diseases. This map illustrates the documented instances of emerging in blue and re-emerging infections in red since 1981 alone worldwide. Points that I want you to take away from this map are that viruses, bacteria as represented by E. coli, for example, on this figure, protozoans like cryptosporidiosis, are represented in this figure. There is no geographic limitation to where emerging and re-emerging infections arise from. And so we have a frequent occurrence that we need to deal with. We're familiar with the first SARS coronavirus that caused an outbreak in 2003. It had a case fatality rate of around 10%. We're familiar with the Zika outbreak that took place about 5 years ago throughout Latin and South America, associated with disturbing images of microcephaly in newborns. We're aware of the high-profile Ebola outbreak in West Africa in 2014 and 15 that caused 28,000 cases and 11,000 deaths, so a 40% case fatality rate. Worryingly, we're seeing that there are new reports that a man who was infected 5 years ago has recently seeded an outbreak in Guinea. So he may well be a carrier of the virus, a phenomenon that we weren't sure existed. And even familiar childhood infections such as mumps have resurged and have even threatened NCAA sporting activities. So clearly an urgent problem. Writ large over all of this is obviously the once in a lifetime pandemic that we're facing. At the moment, we're looking at 130 million global cases and 2.83 million global deaths. So the question is whether we can anticipate the emergence of these infections and the re-emergence of existing pathogens that we're familiar with. So I want to be clear that from now on, what I'm going to talk about aren't pathogens like SARS coronavirus 2. These are pathogens that can efficiently transmit among humans. And so predicting their emergence is tantamount to predicting the event that led to the spillover from an animal reservoir into the human population. And I would venture that that prediction is at the moment beyond us. But I do want to focus on especially re-emerging infections and what happens when that emergence event has taken place. So I want to give you two brief vignettes which relate to how we can use mathematical, computational, and statistical approaches to try and anticipate disease re-emergence. And once that emergence has taken place, how we devise effective means of control and mitigation. Central to this idea of how well a pathogen spreads in a population is this quantity called the effective reproductive number, which no doubt you've read about in the coverage of the COVID-19 pandemic. It's a summary measure that quantifies the expected number of cases caused by a typical infectious person. The critical threshold here is the value of 1. So if R effective is 1, each infectious person only infects one other person, and eventually, by chance, the chain of transmission will break down. But as we think with SARS coronavirus 2, our initial estimates were R effective somewhere around 3. And so what happens is each infectious person will infect, on average, 3 other people, who will infect, in turn, 3 other people, and so on. So that when we look at the number of new cases through time, we see this geometric pattern of growth. And so we're interested in trying to predict when we go from a situation where R effective may be less than 1 to when we cross over this tipping point and R effective exceeds 1 and the pathogen becomes efficiently transmitted in the population. So to illustrate the overall premise of the approach and the actual idea, let me show you a model simulation. This is a simulation of an idealized infectious disease. Here we have a vaccine against the disease, and we're vaccinating newborns as we would against many of the existing charted infections. With the vaccine coverage above the herd immunity threshold of 90% here, year 0, we assume that there's a decline in vaccine coverage, maybe due to increasing vaccine hesitation, as we see for many infections like measles. Concomitant with this drop in vaccine coverage, we see an increase in the effective reproductive number shown here in red, so that by year 15, it hits this threshold quantity of 1. What the model predicts is, as these events unfold, when vaccine coverage was high, we get very few sporadic cases of this disease, so that the surveillance data, as plotted here from the model, shows these small clusters of outbreak that are sparked off when someone from some other population comes in and starts a small cluster of infections. Then a few years after this threshold value of 1 is reached, we get a big outbreak in this simulation. Could we have anticipated that we're about to get this outbreak? That's really the crux of the argument. And to do that, we could measure a bunch of things about these data before the epidemic to try and forewarn ourselves. We could measure, for example, the average monthly reported cases or the variation in the average monthly reported cases. But in this case, I want to show you what happens if you plot the autocorrelation which is a measure of how similar are successive monthly reports. So when vaccine coverage is high, this autocorrelation value is at around 0.5 or lower. But fully 10 years before we have this big outbreak, there's a significant jump, there's a notable jump in this autocorrelation value, and it stays at this high level, almost at 1 from then onwards. So if this jump is indeed an indicator of an impending approach to this tipping point or crossing over the tipping point, we would have a good few years of lead time during which we might roll out a mitigation effort. So we're interested in times when this R effective is below 1, maybe through control efforts such as vaccination, and we want to be able to predict when we're losing control and it's approaching or exceeding this threshold value of 1. And this may happen because there are shifts in population immunity, and I've shown you here a drop in vaccination coverage, but there may be waning of immunity. There may be pathogen evolution to bypass existing immunity, or as I mentioned, vaccine refusal. There may also be a process of host adaptation where the pathogen is becoming increasingly more efficient at infecting the host, so R effective can cross this threshold. So the question is whether we can use, hopefully, some generic kinds of statistical features that we can calculate from surveillance data to anticipate disease emergence. This work has theoretical foundations from the idea of critical slowing down, and was part of a larger project in collaboration with a number of colleagues at UGA, at Penn State, and Michigan. And what I'm going to talk to you about is really work that has gone on in my lab, in particular by Toby Brett. Without going into much detail, we've implemented a machine learning algorithm. I'm going to skip all of these details, but I'm going to point out that this machine learning algorithm has provided us with a pipeline. And this pipeline allows us to take in time series data, so surveillance data of daily, weekly, monthly, whatever, reports of cases through time, for which we can calculate a bunch of early warning signals, such as the autocorrelation function that I showed beforehand, or standard deviation, skewness, kurtosis, whatever. Our algorithm then tells us how to weight each of these measures through time, and then we produce an ensemble of these algorithms into an emergence risk. And we identify a threshold so that when the emergence risk is below that threshold, as identified here in green, we don't predict approach to the tipping point, but when it exceeds the threshold, we have a detection event and we sound an alarm. So let's take this algorithm for a spin. I'm going to show you this idea applied to data from mumps in England. So mumps is caused by a virus. It's part of the MMR vaccine with measles and rubella. The vaccine was rolled out in England and Wales in 1988. And we see that as a result of that, there's very little mumps activity for about a 15-year period. And then in 2005, bam, there's a big outbreak. We also have data for different spatial regions or different spatial organization in England. We have data for all of England, as shown here in the cyan color. We have different regions, the boundaries of which are demarcated with these green lines. And we have local authorities, as shown by these red lines. Here are the data for all of England, presented here in this jaggedy kind of cyan line from 1990 to 2005. I'm also plotting in black this measure of emergence risk that our model has quantified. And through much of this time period, this measure is below some threshold that we've identified in blue, but in the year 2000, this threshold is exceeded. And so we would have a 5-year lead time about an approaching tipping point. We could also look at one of the regions. What happens if we just look at a smaller part of England? In Region 5, for example, again, well before 2005, when the big outbreak happened, we exceed that threshold consistently. If we look at a single small-scale local authority, in this case, local authority 25, again, well into the mid-1990s, we get a crossing of this threshold and a prediction of the approach to this tipping point. We're very encouraged by how this algorithm is performing. It seems to have pretty good predictive power in all the case studies that we've looked at, measles in California, the different serotypes of the bacterium, Streptococcus pneumoniae in Germany, vector-borne pathogens such as dengue in Puerto Rico, or the plague in Madagascar. And the next step, I believe, would be to formally incorporate this epidemiological algorithm within a decision theoretic framework. So the second nugget that I want to talk about in terms of the use of models in our field is really thinking about what happens when you have the disease, when the event has happened and you have the pandemic. So about this time last year, when we're trying to think about how to control COVID-19, there were two ideas that dominated the discussion. One was summarized as suppression. And the idea here is you have control measures such as mask usage, social distancing, isolation. And you want to reduce transmission such that you prevent sustained transmission. And the idea is you want to eventually eliminate the virus locally. So examples of this strategy would be, New Zealand and Australia. Australia hasn't had any deaths since October, and New Zealand hasn't had any since it's had 5 deaths since last June. The second strategy is called mitigation, and the idea here is that you just want to reduce transmission enough so you don't overwhelm the healthcare system, but you want to keep transmission going so that you achieve herd immunity. And this is an idea that was being quite aggressively pursued by the UK government, which is why we wanted to explore it, because it seemed infeasible, at least to us. So we developed a model. I'm not going to go through any of the details of the model. We write down some equations. We take into account the age assortative pattern of mixing among individuals in a population. We take into account how to implement social distancing as a function of age, self-isolation in the model, where these social distancing measures are being implemented, whether at home, at work, at school, and different combinations of these approaches at the same time. But irrespective of the details, the point that I want to make here is that we look at, for example, self-isolation. So how effectively you can identify and remove infectious individuals from the population. And the more efficient you are in doing this, you move to the left in this figure. What that does is it reduces the effective reproductive number of the virus in the population from a value of close to 3 to start with. And eventually, as you have enough self-isolation, if it's rapid enough and compliant enough, you go below this threshold value of 1. On the y-axis, we're plotting the peak hospital burden. So with little control, you're overwhelming the hospitals. This is on a log 10 scale. So you would have at the peak 1 million people requiring medical treatment. And the model allows you to, for example, say what happens if we have the self-isolation and we have no contact among individuals who are 60 and older. So to the left of this point, we achieve suppression and we can eliminate the virus locally from the population. But we also know that there is a hospital capacity in England, which at that time was about 18,000 beds, 17,800. So for the mitigation approach to work, you don't want to cross this threshold of 1 because you want to keep transmission going, but you also don't want to overburden the hospitals. And so you end up trying to design and modify your control strategy. So you're spending most of your time in this very narrow band of the effective reproductive number. So this is effectively the mitigation strategy. And the truth of it is we would not have enough information either about the efficacy of our controls or about the amount of transmission in the population to implement successfully a mitigation strategy. So our work and others led to a general recognition that the herd immunity strategy is going to be tricky to implement, and even the Swedes who were championing and implemented this strategy eventually had to back down and revise their efforts. Ongoing work at the moment locally uses reinforcement learning to identify the optimal strategy for controlling the COVID pandemic. In colleagues in ecology and public health, we've been looking at other non-pharmaceutical interventions such as certification programs and so on to control COVID. And now we're working actively towards an end game strategy for COVID-19 in terms of how we vaccinate in the presence of vaccine hesitation, which seems to represent something like 22% of the population in the US and could be a major barrier. So with that, I would like to thank a wonderful and very talented group of mentees that I've had at UGA for the last few years, without whom I wouldn't be here. With that, I will thank you for your attention and turn it over to Diane Marie Amann.
Thank you so much, Professor Beach, Simmons, and Rohani. I am delighted to bring up the final lecture in this series, and it's going to be something a little bit different. If I may share my screen, I will go forward. My lecture is focusing on research that I've been doing for a number of years now on Nuremburg Women. That's the title of the book that I am writing at present under contract with Oxford University Press. And with the great assistance, I want to thank my Dean, Bo Rutledge, my many colleagues at the University of Georgia School of Law and its Dean Rusk International Law Center, as well as the School of Public and International Affairs, at which I'm pleased to have a courtesy appointment, and indeed the African Studies Institute, where I'm an affiliated faculty member. The research has been supported by at least 3 fellowships from the USC Shoah Foundation, the Bonnevero Institute of Human Rights at Oxford University, and the Max Planck Institute in Luxembourg. And so what I'm going to do today is give you an overview of the book, which will focus on the 6 women that you see here. They represent a multinational cohort of women professionals from the United States, Great Britain, France, the Soviet Union, and one woman who was a Jewish-born German, or a German-born Jewish woman who fled Germany just before the Germans invaded Poland, fled to the United States, and returned to Nuremberg several years later as a naturalized US citizen. So that is my core database, if you will. The subtitle of the book is New Histories of the Monster Trial. That is a phrase that I pulled out of contemporary feature stories that were written by a journalist named Elsa Triolet, who went to Nuremberg during the first major trial, the one that included the 4 victorious Allied powers. And she observed proceedings for the better part of a couple of weeks, and then wrote lengthy articles about what she observed. Her title is The Waltz of the Judges. And I've circled here her reference to Le Procès Monstre, the monster trial, where she says that she had been overwhelmed by the existence and the work of thousands of people in connection with the monster trial. And for me, that's been a really wonderful metaphor, not only because there were monsters on trial, not only because the crimes of the Nazi regime were monstrous, but also because the project itself was a monster of a trial. 4 countries plus a dozen other countries assisting, as Triolet says, thousands of individuals involved. The first trial went for a full month, a full 12 months, a full year, and was succeeded by 12 additional trials that went from 1945 all the way through 1950. The image of monster also is striking to me because there is, in fact, a monster in courtroom 600 at Nuremberg. This is a bronze mask that hung over the courtroom throughout the year-long trial. It is believed to be an image of Medusa, the woman with the head of snakes that strike evil. And this has been striking to me in my work because she also turns out to be, through reinterpretation over the years of works going back to Ovid, a bit of a feminist icon. And so the story of the Medusa and her experiences with men in some ways is an inspiration and a reminder to think through what the existence and work experience would have been of the women that I am interested in. Now, by this point, you're probably saying, wait, what? There were women at Nuremberg? I must confess that as late as 2007, although I have been working in the field of international criminal law for years, I myself was unaware that there were women professionals working at Nuremberg at the first trial or any of the trials. And that is in part because of images like this panoramic picture of the trial. They are reflective of most of the literature. There are no fewer than 40 book-length memoirs written about Nuremberg. Only one of them was written by a woman at the first trial, and it happens to have been published in the original Russian and is available only in German. Almost all of the rest are either in English or French and written in very celebratory ways by the males who led these affairs. If we look at this picture initially, we see sort of a gray tedium of the day in and day out year-long trial. Lots of gray-haired folks, a lot of them perhaps balding. And we think of it then as an all-male affair. If we look closer, however, we discover that in this image, there are no fewer than 20 women present on just an ordinary day. The women at the very top, the one on the far right, by my reckoning, is the only one who might have been just a casual observer of the trial. Every other woman is dressed professionally and seated in a place suggesting that she was at work at the time. The women in sort of the middle cohort of the room are all journalists. There are many, many women journalists of many nationalities who covered the trial day in and day out, or for brief occasions, as with Triolet. One of the women in the room at this time goes on to become the first woman ever to win the Pulitzer Prize on account of her reporting of the Korean War. And then in front of the bar, these are the women professionals who helped run the trial. They include analysts, interpreters, and indeed at least 2 women lawyers. The one on the far right is the French woman lawyer on whom I'm focusing, and 2 over, the sort of blonde woman in the middle, is one of the US lawyers. Now, it would be easy enough to think about the story of women at Nuremberg from a women-only lens. But perhaps the most significant for me revelation in the work that I've been doing, and I should say my database includes not just the 6 on whom I'm focusing, but well over 200 women who worked at the trials in different ways. Their status or identity as a woman was often not as important than other identities. And we shouldn't therefore try to create an ideal woman or a proto-woman at Nuremberg and invest in her all of our experiences. And in that insight, I draw from an essay by a French theorist, Hélène Cixous, who refers to the Medusa again and says there is not a general woman, a typical woman. We cannot speak of one feminine sexuality that is uniform and homogeneous. Another theorist, Karen Knopp, who is a professor at Toronto, says with regard specifically to this kind of experience, women's identities are multifaceted, and on certain issues, they may choose to participate in international law in ways that reflect their identity as a part of an ethnic, linguistic, or religious group, rather than the united front of gender. And so indeed, what I have found is that in addition to identities as being female, as being women, one's race, ethnicity, national origin was extremely significant and affected experiences and participation, both participation that folks wanted to do and participation that women were permitted to do. Sexuality and gender expression, marital status, all of those played a role. Political ideology and other beliefs, whether one was Jewish or of another religion, whether someone was identified with what we would now call the New World Order of liberal democracies that emerged after World War II, or conversely was a communist, either a member of the Soviet Union and the Soviet bloc, or not infrequently members of the French delegation, and indeed before World War II, some members of the US and the American delegation. And then finally, class issues, socioeconomic status, educational status is profoundly important. And so it's the intersection and interplay of these identities at one of the major projects of the mid-20th century that is essentially what I'm studying. The work is a work of legal history, of course, because I'm looking at this important trial, and it fits within a trend in international law, what some have called the turn toward history in international law, a looking at history, and indeed, very, very importantly for this project, a looking at histories from below. All of my subjects were subordinates. None of them was a leader. They were always in a relationship with the hierarchy. And so the work sort of is part of that stream as well. And so what I do then is analyze within these intersecting identities and these varying experiences that come with them, starting from the left, Betty Richardson, who was British. She had spent World War II as an intelligence officer in the Women's Royal Naval Service, intercepting U-boat signals. And after the war, she is tasked with several other Wrens, as they were known, to go to Nuremberg and use their skills to help in the case against the German admirals. To her right is Tatiana Stupnikova, who is the woman who wrote the only memoir by any Nuremberg woman in the first trial. She was a veteran of the Red Army and had spent the liberation going with General Zhukov from camp to camp, from battle to battle, and interpreting from German into Russian for him. She becomes one of the first interpreters. Indeed, simultaneous interpretation was invented at Nuremberg. And so she was one of the pioneers. Interestingly enough, she was not, in fact, a communist. Her mother was in a gulag as a dissident at the time that she herself was assigned to Nuremberg. To her right is Harriet Zetterberg, who is an American who was #1 in her class at the University of Wisconsin Law School, the first woman editor-in-chief of that law review, and went on to be the first woman ever who was a Sterling Fellow, postgraduate fellowship at Yale Law School. She is the only woman who is in the trial proceedings frequently throughout the trial, and indeed is seen here helping to present one of the cases. Below her is Dr. Aline Chalouffour, the sixth woman in history ever to receive a doctorate in France in public international law. She is the only woman on the French delegation. To her left is Edith Simon, who was the German Jewish emigre whom I mentioned. And then at the far left is Catherine Feit, who was a State Department lawyer, another American who helped draft the Nuremberg Charter. Adding to their stories and adding to the fabric of my project will be cameo appearances, if you will, of other women who begin to really complete this discussion. In addition to the lenses that I've identified being important for my 6 women, they became particularly significant with other women. So below right is Erika Amann, the daughter of Thomas Mann, the novelist, who comes to Nuremberg as a journalist. She lives openly with another woman throughout that proceeding, as did several other female journalists. Sexuality was very much present in a way. One commentator said the sexual revolution was all go at Nuremberg. Americans would have been shocked back home had they known. And so her experiences will be of use as well. And then finally above her is Philani Ahuna, who was born in Honolulu to parents who were Chinese and indigenous Hawaiian. She makes her way to Nuremberg as a court reporter. And I just ask you to imagine what the experience would have been for someone to be perhaps the only person of Asian ancestry, an object of curiosity indeed by Nuremberg defendants here, like Hermann Goering, whom she is very quite close to, public enemy number 1 in the trial, at what was in fact a trial about race and the worst kind, the most extreme kind of racial discrimination. So that's what I'm going to be looking at. I thank you for your attention, and I am happy to speak more about anything in this project. Thank you very much.
Thank you, Professor Amann, Professor Rohani, Professor Beach, and Professor Simmons for the very, very interesting presentations. I hope the audience will get a sense of the breadth and excellence of UGA faculty research and scholarship. I will now invite everyone to post questions through the Q&A feature. I will try to do my best to direct the questions to the right individual or the panels as a whole. To begin with, I would like to ask a question, knowing that the pandemic is high on many people's mind. So ask the first question to Dr. Rohani. So what are some of the key lessons that infectious disease researchers and experts have taken from the pandemic?
That's a complicated question. And I don't have a single answer for that. I would break down my thoughts into this pandemic has identified scientific and methodological gaps in our understanding and in our ability to predict. And in particular, it's identified the fact that we're not very good at predicting human behavior and human decision-making and incorporate these such phenomenon into our models for forecasting. I think it's also identified a shortcoming in the way that we communicate science to the public, in the sense that we typically resort to fact and information, as is our wont, and it appears that other actors maybe are very successful by appealing to emotions instead. In terms of policy, I would say that our research can potentially be impactful, but it's really an implementation issue that we need to be more proactive in. And so I think the US, for example, needs a single coordinated organization for formulating strategy based on best available evidence and a plurality of disciplines. And I think this was particularly successful in places like South Korea and New Zealand. And then one thing that's close to my own heart is I think we need a federal institution that really spearheads modeling and forecasting for infectious disease preparedness and control. It's a surprise that we don't have such an agency. And I'll point out that whenever we have whenever there's a hurricane, we don't expect academic meteorologists to drop everything and tell us where the hurricane's going to go. We have federal agencies for that, and we don't yet have that for infectious diseases. So those are kind of three broad lessons that I would say we have learned.
Okay, thank you. I will now go to the Q&A panel and select some questions for the panelists to answer and to discuss. The first one, I think, is direct toward Dr. Simmons. Are there any other chemical processes other than methylation that may impact aging?
So there are a lot of the body responds to stressful situations like threatening conditions, for example. The list of things that enhances aging that I reviewed for you were things like discrimination and hostile relationships with friends and neighborhood threat and economic hardship and so on. All of those things are impacting the body in a wide variety of ways. They're impacting the sympathetic nervous system, the HPA axis. They're jacking up your cortisol. They're driving up. They're affecting your immune system and driving up inflammation. All of those things have health consequences, but they're also impacting your this more general measure of epigenetic aging. So epigenetic aging certainly is not divorced from these other biological processes. As a matter of fact, it's very much a part of it. It's because ugly events ugly circumstances tend to impact such a wide variety of biological systems that these things spill over and are affecting you systemically in terms of your overall aging. I don't think I answered that very well, but that's as good as I can do right now.
Great effort. Now, a question for Professor Amann. Professor Amann, your project focuses on the role played by women at Nuremberg, who, as you observe, typically occupy the subordinate positions. By contrast, today, the chief prosecutor of the ICC, Fatou Bensouda, is an internationally accomplished female lawyer of Gambian descent. Can you speak about your experience advising her and drawing on your historical study of Nuremberg, your observations of the ICC working under her leadership?
Well, thank you, Provost, too. Yes, I've had the privilege since 2012 to serve as the special advisor on children in and affected by armed conflict to the prosecutor, Fatou Bensouda, who in fact visited the law school a few years ago and gave a keynote lecture. It is true that she is in a leadership position. It's also true that international criminal law attracts far more women than many might think given the gruesomeness of the kind of issues that we're dealing with. That said, the erasure of the women whom I'm studying right now, and I sometimes, when I play that trick with the photo, I sometimes refer to them as the visible invisibles, or the invisible visibles, whichever you want to say. The fact that nobody knew they existed, that Harriet Zetterberg wrote the brief on Jules Streicher and other leading defendants, but the man who's standing at the lectern read it for her because no woman was permitted to speak, even if they were admitted as a lawyer to the tribunal. The erasure of that has made the experiences of women like Prosecutor Bensouda and other leaders in international criminal law more difficult than it should have been. They've had to blaze a trail, not understanding that the work had already been done for them. And so when I've given this presentation at the International Criminal Court, it has been truly eye-opening even to the people who are working there, and leave all of us with the feeling that the profession will become stronger by understanding more accurately its historical roots. So thank you for that question.
Very good. Maybe I can ask a follow-on question to Professor Amann. What was the role of the female American lawyer in the trial? Specifically, was she in a supporting role on the trial team, or did she provide critical legal analysis or arguments?
So there were two American lawyers, Catherine Feit and Harriet Zetterberg. Feit helped draft the charter, the governing document, and was very significant because she was the only international lawyer doing that project. Most of the rest of them had come from other spaces like criminal law. And so her insights into international law were quite important. Zetterberg is there day in and day out, building the case against at least three of the principal defendants. And that included driving in a Jeep to Czechoslovakia to interview witnesses and bring them back to Germany, doing legal drafting, etc. The third woman lawyer was Dr. Chaloufour, who in many ways may have had the most significant role. She had been in Paris right after the camps were liberated, working for the Ministry of Justice, and she was essentially the intake lawyer when detainees were coming out of the camps, including one fellow named Francesc Bush, a Catalonian who had smuggled photos out of the Mauthausen camp. And they became one of the central pieces of evidence in the trial. And it was she who had met him, took that evidence, and helped build that part of the case.
Yeah, thank you so much. Next question, I think, is directed toward Dr. Simmons, but I believe Dr. Beach can also participate in the discussion. If I understand, your research shows that marriage increases lifespan. Is that true for both sexes? My understanding has been that men benefit from marriage while women suffer detriments. What does your research indicate?
I will let Steve go first because he is the real marital expert on this project.
We generally find that it's not marriage per se that affects people's health and well-being outcomes. It's the quality of the relationships that they have. When we focus on trying to do something that's going to benefit people, what we try to do is help them improve the quality of those relationships, whether they're married or not married or in some other kind of relationship. And that's what seems to matter. So Ron, I don't know if you want to build on that.
The only thing I'll say is that there is this lingering notion still, okay, it was first promulgated during the 1970s, that women benefit from romantic relationships and men don't. And that just has not turned out to be true. Or maybe it was true back then, but it simply isn't true now. both respond to a high quality relationship.
Okay, thank you both. One more question for Professor Amann. With the percentage of female partners at large law firms or federal judges at international courts and tribunals still hovering below 20%, how has your research of the role of women at Nuremberg informed your view of the trajectory of women in law?
Well, it is slow, and it's not entirely clear that the curve always goes upwards. At the time that these women were practicing at Nuremberg, only 4% of the entire bar of the United States were women, not law professors, not law partners, 4% of women. And that just gives us an idea of how extraordinary and true they had to be the top #1 person in their school to get these kinds of jobs. The work is constant. And I think what we learn if we look over time that reinforcing people's understanding of the contribution of what I call the other others, women, people of color, gender minorities, etc., is important to continue what is a project still for us today of building diversity in a genuine and sustainable manner, not only in the legal profession, but indeed in all professions.
Thank you. Maybe one more question for Dr. Simmons and Dr. Beach. What is an immediate step that individuals can take to have a positive impact on their well-being?
Go ahead, Steve.
Well, I'm going to say that there is nothing quite like investing in your family to yield immediate dividends. And that's something that our research supports. Ron?
Yeah, I would say anything that makes you enjoy life, feel safe and secure, as opposed to all those things that age you, which are threatening and scary and amp up depression, anxiety, which means social relationships. Since we're social creatures, social relationships are awesome. So involvement with other people, and good friends, people who make you feel safe and good about yourself. All of those things are going to be excellent.
Okay, thank you both. And I want to thank our Regents professors for the very insightful presentations. So we have come to the end of our time. Again, thanks to the Regents professors for the presentation. We greatly appreciate your effort in terms of your scholarship and dedication to teaching and dedication to UGA. Thank you, President Moorhead, for being here. And it's a great pleasure to have you speak to all of us. Of course, we want to thank everyone in the audience who joined us for the charter lecture. I hope you will join us in future such lectures. Have a great day. Thanks again.